pleased to introduce the first speaker. I'm Ken Sharp, by the way. I'm going to be the moderator for this session, and it's my pleasure to introduce our first speaker, Jack Scannell. Jack worries about some of the big problems involved in discovering new drugs, not just the scientific problems, but the economic problems. He was trained in physiology and medical sciences at Oxford and at Cambridge. He's worked in the drug industry. He's worked as a consultant. He's a fellow at Oxford Center for the Advancement of Sustainable Medical Innovation. And today, he's going to help us solve a problem, a puzzle. There have been spectacular improvements in research techniques in the drug industry in recent decades. Yet drug discovery has become less efficient and more expensive. And Jack's going to tell us how come.
We've had a day and a half of fantastic, uplifting talks. It's a nice warm day outside. You've had a lunch, feeling a bit sleepy. And now, for the next 45 minutes, someone who hasn't worked in a lab for 17 years is going to use statistical decision theory to tell you why all the biomedical scientists in the room are doing a lousy job, right? Down the compass, full speed ahead. The title is a misquote from an order given at the Battle of Mobile Bay by the wonderfully named Rear Admiral David Glasgow Farragut. One of his boats, probably that one, struck a mine, which at the time were called torpedoes, keeled over and sunk. And rather than retreating, Farragut ordered, "Damn the torpedoes, full speed ahead." And I decided to misquote Farragut as a title to my talk, because I think when it comes to drug R&D, full speed ahead is the sensible thing to do only if you're very certain that you're headed in the right direction. If you're not certain you're headed in the right direction, if the map is not accurate, if the compass is not pointing in the right direction, full speed ahead gives the simulated impression of progress and furious activity, when in fact all you're doing is heading in the wrong direction at great speed. And the analogy that I think I'll use here is that I think of drug discovery a little bit like trying to find a small island in a large and rough ocean. And in that navigation task, the quality of the map and the compass may actually be much, much more important than the horsepower that is deployed moving the boat along. Okay, so this is a caricature of how drug discovery works, but it's not my caricature, it's the drug industry's caricature. So if you ask the major trade associations or the drug companies to explain R&D to politicians or policy people or the masses, this is what they say they do. And it's based, I think, on this logic. It's based on the idea that there are lots of identifiable little pieces of biological machinery, often coded for genes, usually proteins, that either behave or misbehave in a certain way, and that somehow causes problems with a particular disease. Maybe it causes Down syndrome, maybe it causes male pattern baldness. There's a whole, but some molecular component misbehaves, and it causes a disease phenotype. There is then the assumption that that molecular component can be selectively, its behavior can be changed by giving the patient a molecule, often a small molecule drug, which they take orally or maybe something injected. That thing then sticks on to the molecular component that's either behaving or misbehaving. It changes the way it behaves. It causes an improvement in the disease phenotype, and it does it without unacceptable toxicity or side effects. Presumably, one believes there are still many identifiable, exploitable instances of molecular components that have gone wrong, drugs that we haven't yet used that we could, and disease phenotypes. And if you believe all these things, this industrial process would be an extremely efficient way of bringing lots and lots of new drugs to market at low cost. And a good chunk of the $150 billion roughly a year spent on pharmaceutical R&D by governments and by the drug industry is spent doing this. Certainly not all of it, but a lot of it is spent doing this. So We identify little pieces of biological machinery that have gone wrong, often using genomic technologies. We get large compound libraries, sometimes computationally, sometimes in real life, and we test them against those little bits of biological machinery. We do a bunch of clever stuff to give them the right characteristics to be products. So, for example, coming back to the example of male pattern baldness, you might think you've got a wonderful drug to cure male pattern baldness, but if it can be only delivered by an intracerebral injection, it's unlikely to be commercially successful. So you do a bunch of stuff so it can be formulated another way. And then if you're convinced that it does what you think, and it doesn't give rats and dogs cancer, and it's not overtly toxic, the FDA and your investors will allow you to conduct a bunch of clinical trials. If we go to those gray arrows, which show the activities you do before you get to clinical trials, the scientific success in making those things better has been truly spectacular. We know that sequencing DNA is very important for identifying drug targets, for example. That, and this is out of date now, when I checked, probably in 2012, it had got about 10 billion times cheaper and more efficient than in the very early 1970s. Maybe it's 100 billion times cheaper now. X-ray crystallography, just to give another example, another technique, is a way of identifying the structure of the little bits of biological machinery that have gone wrong so that you can work out what kind of drugs are going to stick to them before you even have to do any wet experiments. And this has got maybe 10,000 times cheaper, and we now have large databases of the structure of proteins so that you can work out what kind of drugs are likely to work on them before you ever do any expensive wet science. And I could say the same about a whole bunch of other technologies, and we've got animals, for example, that we can make, give genetic changes so they try and mimic the disease in man, and we can test our drugs in those before we put them into human trials. So one would expect, given the way the drug industry says drugs are discovered, and given the huge improvements we've had in all of the technical inputs that most people who do drug discovery think are important, you would have expected the process to have got a lot cheaper and easier. That's not the case. This graph is the number of drugs brought to market per $1 billion industry R&D spend from about 1950 to probably last year or the year before. It's a logarithmic axis, right? So each one of these units is a 10-fold change. In 1950, when we didn't know anything and we didn't have any good drug discovery tools, we seemed to be able to discover about 100 drugs per $1 billion of R&D spending, inflation adjusted. This halved roughly every 9 years. We have had a little uptick lately, which I'll talk about later. And you can see there are some obvious effects here. So, for example, regulation. After thalidomide, before thalidomide, you didn't have to show your drug worked. Right? You just had to show it wasn't toxic. Showing drugs worked clearly depressed the number of drugs that were discovered for a few years, but things recovered. Here, the FDA cleared a bit of a backlog. That made a difference. Interestingly, the first wave of biotechnology didn't seem to make much of a difference on the trend. Again, I'll come back to this later because I think this is a real uptick. But even this uptick only takes us back to the kind of glory days of 2004, right? The big effect is that it's gone down roughly 100-fold. This has real implications. I used to work in the UK equivalent of Wall Street, and my professional opinion when I was an equity analyst was that the average big drug company was losing money on R&D, right? So although drugs are eye-wateringly expensive, and so expensive that many health systems can't afford them, I was of the view that the cost of R&D was such that the average big drug company was essentially only generating maybe 90 cents on every dollar it was spending on R&D. Now, of course, if you spoke to the drug companies about this, they'd say, yeah, that's true, but we're not average. None of them were. But this is a policy problem, right? Because if private sector funds about 2/3 of drug discovery, if the private sector starts thinking, actually, this is not such a good business to be in, right, we see an acceleration in the rate with which laboratories are shut and medicinal chemists are made redundant, and we just get less drugs discovered. And it probably doesn't make the drugs, and the drugs that are discovered will remain very, very expensive. So I think this is an important public policy problem. And you would imagine, as an important public policy problem, there's a huge body of highly authoritative, quantitative work that has tried to work out exactly why we face this particular situation. And if you imagined that, again, you would be wrong. There are a few souls who work on this problem. There's Richard Barker, who's sitting there, who is one of them. I'm one of them. But it's a relatively small band of people who are interested in how it is that all of the inputs can get so much better while the output gets more expensive. just to depress you a little bit more before I maybe get marginally more optimistic, I had a look around for other industries where the inputs seem to get more efficient, but the outputs seem to get more expensive. And I first of all thought, well, oil industry, obviously oil must have got more expensive over time because we've sucked all of the easy oil out of the ground, and now we're drilling oil wells in deeper and deeper water, further and further underground. In fact, oil, in real terms, is cheaper than it was probably 100 years ago. The same is true of lots of other things you dig out of the ground: nickel, zinc, lead. Lots of things seem to get cheaper over time. One thing that didn't get cheaper over time was whalebone. Right? It got more expensive over time, despite the fact that the boats that you could use to go and hunt whales got much more efficient. So this is the cost of driving your ship around the ocean going down. The price of whalebone, surprisingly, going up. And here's the reason. This is the number of whales that were caught between 1800 and 1880. And what you see is that here, you have a kind of resource depletion problem. Right? So are we facing a resource depletion in the drug industry? Have we run out of something? And if so, what is the something we've run out of? So again, when I was an equity analyst, you have remarkable access to the people who run drug companies, and you can ask them, usually only two, but you can ask them two impertinent questions at meetings, because everyone has to get a turn answering questions. And the question I generally asked them was, why is it that all of the technologies you have to discover drugs are so much better than they were 50 years ago, but it costs you 100 times more? And this was the explanation they typically pointed to. It was a kind of low-hanging fruit explanation. So this chap is sitting, he's a hunter, and he's sitting with something called Tasmanian tiger. And it doesn't matter if you had high-throughput Tasmanian tiger hunting technology these days, because the last Tasmanian tiger we know about died in 1936. Right? So this is a kind of set of resource depletion arguments. And the typical drug industry response, or senior management response anyway, was a sort of shrug of the shoulders and say, well, we've picked the low-hanging fruit, which I always thought, although never said, because I was too polite, was not a terribly good explanation for someone deploying $1 billion a year on R&D, because the fruit ain't going to get any lower, right? So if you really think it's difficult because you've done the easy stuff, you should probably stop what you're doing. There is another set of explanations, which I would say is to do with the progressive adoption of worse discovery methods. Right? So this chap is trying to transmute lead into gold. It doesn't matter if he builds a turning lead into gold omics facility. He's still not going to be able to turn lead into gold. Right? So these are the two broad classes of explanation that can explain why all of the inputs seem to get much more expensive, but get cheaper, but the output gets 100 times more expensive. And the profound problem, I think, for anyone working in biomedical research is, the more preposterous you think either one of these is, the more you have to agree with the other one. Right? So if you think, actually, it's ridiculous, we haven't run out of stuff to discover, then you're kind of forced to conclude that most of the money being spent is spent on stuff that we shouldn't be spending money on. Or if you think, actually, we're doing a brilliant job and there's no one doing this lead into gold stuff, then we really are in trouble. Right? So, again, me and a few other people, not a huge number of people, have taken interest in this and published a variety of reasons. You won't be surprised to hear there's not one reason, right? It's a number. Regulations got much more difficult. There's another particular problem in the drug industry, which some people call the best in the Beatles problem. So the drug industry is an intellectual property business, but it's unusual in that people don't get bored of the intellectual property. So imagine how hard it would be to get a new record sold if every new record had to be better than the Beatles, if you could download all of the old Beatles records for free, and importantly, if no one ever got bored of listening to the Beatles. Right? If you had those three criteria, you wouldn't get many new records. But that's exactly what you get in some therapy areas. So for example, metformin in diabetes is a bit like the Beatles. It's incredibly good, it's incredibly cheap, and people don't get bored of taking it. Right? So you have this ever-improving back catalogue of free stuff that makes it harder and harder to bring new things to market. In most intellectual property businesses, people get bored of the old stuff and they want new stuff even if it's no better. Right? So there's a number of particular problems in the drug industry. But I'm going to focus on one which I think is a mixture of resource depletion and the progressive adoption of worse discovery methods. And in a sense, the resource depletion problem has pushed people to do this. And then at the very end, I'm going to suggest what might be done about it. So I said at the start there's going to be a bit of statistical decision theory. This is where it starts. I have to kind of think about the drug discovery process in a slightly formal way. And there's a term I'm going to use, which is model, screening or disease model. And this slide is really to remind me to explain it. Because if I don't explain it now, I think what follows will be incomprehensible. So let's imagine you're trying to discover a new anti-cancer drug. What you can't do is take millions and millions of random compounds and try them out in patients, right? Because it's both unethical and very expensive. So what you do is you, first of all, you get a little bit of biological machinery that you think is the bit of biological machinery that's causing the patient's cancer, and you put it in a dish. And then, because you can afford to, you typically test a very, very large number of compounds against that little piece of biological machinery to look for things that stick to it and may alter its behavior. And then you test them maybe in cells in a dish. And the ones that do best in cells in a dish, you maybe test them in rats, which have the cancer because you've genetically modified them. And then the 2 or 3 that seem to work in the rats, you may test them in people. So you have this kind of filtering process where you start with lots and lots of molecules that might perhaps be a cure, and you use a series of tools to measure them, and you think the output of those tools somehow correlates with potential use in man, and each time you throw away 90% of what you tested, and you just take the best 10% and you put it into the next step. So each one of those things where you test and rank a bunch of candidates and discard some is what I call a model, a screening or disease model. And you do it because you think the rank ordering of things on your screening or disease model somehow agrees with the rank ordering you would get if you could afford and get legal permission to put them all into people. So here we are, compasses and speed. This chart illustrates actually most of the basic ideas, so I may spend a bit of time on it. Here we have a good compass, here we have a bad compass. On this axis, imagine you had a large collection of potential drugs, millions of potential drugs, you don't know which are good and which are bad, and you test them all in your screening or disease model. So this axis represents the score that each of those drugs would get on your screening or disease model. This axis represents the score that each one of the drugs would get if you really gave it to a human patient. And of course, you're never going to give them all to a human patient because you can't afford to. But what you hope is that your screening or disease model gives you roughly the same answer, or roughly the same rank ordering of answers, as you would get if you actually tested them all in a human. And this is a good compass because what you see is that in general, the score you get in your model is pretty close to the score you get in a human. And the color coding here just represents the probability that you'd find a molecule in a particular region. If we go back to the previous slide, you see the same here, right? So here we've got a model which is looking at how well a molecule, each point represents a molecule, a drug molecule, sticks to a particular protein, and this is how well the molecule inhibits the activity of this protein. Right, so here we might be saying, well, we want molecules that stick to the protein really hard because we believe that's going to help us predict which molecules inhibit its activity. So here, this is how well the molecule scores in the model. This is how well the molecule scores on the important dimension. And I've drawn some lines on this chart. This line represents the point at which you say, OK, it looks good enough on our model that we're going to put it into clinical trials. And this line represents the point at which the FDA says it was good enough in clinical trials that we'll let you sell it as a drug. So what you can see here is we've got a bunch of true positives. These are things that our R&D process said should be drugs that were drugs. Here are things that are false positives, things that our R&D process should be drugs that weren't because they didn't cross the FDA threshold. And here we have false negatives, things that our R&D process should have picked up but didn't. And the last bit of definition is here. Here's the speed. As you go from here to here, molecules get rarer and rarer. Most drug candidates are pretty lousy. You've got lots of things here which don't work in your model and don't work in people. Right? That's the normal way. So let's suppose only 1 of 100 of your molecules would work in a human. Right? If that's true, you have to set your threshold at a point where you're going to capture 1 out of 100 things, and that means on average you have to search 100 molecules to find the 1 out of 100. If you only think 1 out of 1,000 molecules will work in a human, you have to search 1,000. If you only think 1 in 10,000 molecules will work in a human, you have to search 10,000. So what brute force power, or what throughput does, is it lets you search further and further out on this axis And if your model is a good predictor of what happens in man, it lets you identify things that are likely to be better and better and better in humans. So this compass, on the other hand, with this model, you've actually got a rather low correlation with what happens in man. So imagine what happened if we applied these two models and then did clinical trials. In this case, the amount of stuff in this quadrant, true positives, is relatively high compared to false positives. There's only a little bit here. There's quite a lot of mass here. So most of our clinical trials would succeed. If we use this model, there's lots of stuff here and not much stuff here. So the vast majority of our clinical trials would fail. OK, so enough decision theory for now. Let's talk about something that matters a bit more. Antibiotics. The drug industry has had a pretty poor record in bringing new antibiotics to market over the last few years. And although the commercials of antibiotics don't look great, it isn't entirely through want of trying. A number of big drug companies between about 1995 and 2005 made a huge effort to discover antibiotics. Glaxo wrote it up in great detail, but a number of other companies tried to do the same. They didn't write it up in great detail. And one of the reviews of the depressing antibiotic discovery experience said this, which I'll read because I think it's important. Is it not peculiar that the first useful antibiotic, the sulfanilamide drug Protonsil, was discovered by Gerhard Domach in the 1930s from a small screen of available dyes? So he just went to Bayer, who made dyes, and he said, can I have some dyes? Because I'd like to test them in case they kill germs. Maybe I'm exaggerating slightly. And they said, well, OK, we've got 300 in the cupboard. Give them a go. And he found the world's first useful antibiotic. Between 1995 and 2005, several major drug companies tested around 10 to the 7 compounds from their libraries. And the targets they identified them using this beautiful logic. They said, let's look for genes in bacteria that are essential. If the bacteria doesn't have the gene, it's dead. Let's look for genes which have got no homologs in humans, because those things then won't, any drugs that kill the bacteria won't kill people. And let's look for essential genes that are common across a bunch of bacterial species, because then we'll get broad-spectrum antibiotics. And then we're going to test those against our enormous 10 to the 7 compound libraries. Poor old Domach just gave a couple of hundred dyes to a few mice that he'd infected with bacteria. He found something these folk didn't. Is it not peculiar? Well, the next slide, which is essentially a mathematical derivation of the previous but one slide, shows it's not necessarily peculiar. What we have on this axis is the quality of the compass. This is the degree to which your disease or screening model output correlates with the outcome of interest in man. So 1, perfect correlation, 0, no correlation, right? So we've got a great compass over here. We've got no compass at all over here. This axis corresponds to your brute force power, the number of compounds you are able to screen. And it's a logarithmic axis. So here you can screen, let's say, 10 compounds, 100 compounds, 10 to the 3,000 compounds, up to 10 to the 7 compounds. And here we've got Gerhard in the 1930s and the global biopharmaceutical industry between 1995 and 2005. So if it was the case that the results in mice, infected mice, are very highly correlated with the results in infected humans, whereas the 10 to the 7 compounds tested by the drug industry were tested in models with a very low correlation with outcome in humans. The ratio of true positives, i.e. things that really work in clinical trials, to true positives plus false positives, right? The total things that your model says test. Sorry, I should have explained that. So the color coding here, apologies, is a thing called positive predictive value, which is the true positives over all positives. So it's the proportion of things you put into clinical trials that work, effectively. So this is good, this is bad. So here we have Domach, model very highly correlated with the human clinical outcome, only testing a couple of hundred molecules, higher positive predictive value than a very poor model where you've tested 10 to the 7 compounds. And this chart was simply done assuming that things that will work are really, really rare. And this chart was done assuming that things would work or things that would satisfy the FDA are about 10,000 times or are much more common. And so So the point here is that the DOMAC result isn't surprising if you've shifted from highly valid screening disease models to much less valid screening and disease models. And I'll make two more points about this chart. One, if you look at the direction of contours, what you really want is you really want things that are going to get you from the very dark regions of the chart to the very light regions of the chart. So how do you go from dark regions of the chart to light regions of the chart? Well, one thing you do is you start with a model that's really, really good, which is very highly correlated with the human output, and then you make it faster. Alternatively, you start off with huge brute force power, and then you somehow make your model more valid. But one thing that really doesn't work is you start with a lousy model, and then you just make it faster. Right? There is no point doing high throughput nonsense because you simply generate lots and lots of false positives. So, down the compass, full speed ahead. Here, I think, is the result of the decision theoretic analysis, and then I'm going to briefly say what might be done about it. So, the ability of the drug discovery process to identify things that are going to work in man is exquisitely and surprisingly sensitive in quantitative terms to the validity of screening and disease models, i.e. the extent to which their output correlates with the output in man. And roughly, for those of you who are sort of practicing biologists, an absolute 0.1 change in the correlation coefficient between the output of your model and clinical utility in man is, in many cases, as or more important than doing 10 times as many experiments. Right? How many of us know, even, what the correlation coefficient is between our model and clinical utility in man? And I think most biologists, if they do an experiment where the correlation coefficient is 0.6 and it's 0.7, you'll think, well, really, there's kind of not much difference. Right? That's kind of lost in the noise. So these very, very small, or what would be perceived as relatively small effects can be incredibly important in quantitative terms. And I think this is important because the decline in productivity I showed earlier in the face of massive increases in brute force efficiency, this is a big effect, right? Things just haven't got a little less efficient. They've got much less efficient, and the inputs have got much more efficient. So any reasonable explanation here has to be quantitatively powerful. And I think the model validity effect is powerful enough, 2, 3, or 4 percentage points lost in the correlation coefficient, and you suddenly lost 10, 100, 1,000, 10,000 times worth of brute force power. So, well, why then might the validity of the stock of models available to biomedical scientists have declined? I think one is a bit like the whale hunting problem, right? So there's some models that work really, really well. They tell you exactly which drug is going to work in man, and those are the models that correspond to the diseases we've cured, right? And scientists and drug companies don't like working on all the diseases we've cured. So you retire all the models that work, right? So there's this universe of models, and we simply deplete it of the ones that work, and we're left with the ones that don't work, the ones that have low validity. So we keep turning our wheels, testing molecules. The therapeutic opportunities that remain are those therapeutic opportunities associated with bad models. That's one problem. I think certainly, and I think this is probably where I'd be more controversial, but certainly the first wave of industrialized target-based drug discovery, I think, is now widely recognized as having suffered from real problems of validity. Right? So people would just test stuff in a way that was fundamentally non-predictive. And again, my sense is that what is academically fundable is not necessarily that which has high validity. Those two things are not necessarily congruent. And I think there's a third sort of more managerial thing here that may apply more in the industry and less in academia, which is it's very easy to manage people on how much you've done today. It's very hard to manage someone on vague qualitative views. So actually, until you've got 5 drugs discovered in the therapy area, no one knows whether the models are any good or not. So you can say, well, how many molecules did you screen today? Right? How many things did you test? People can answer that question, and you can manage it. But what's your gut feeling about the model? Is it good or bad? That's a much more difficult thing to manage and measure. So my sense it may have got squeezed out a lot of industrial management. So what can be done? How can one improve validity? This chart I actually came across, or this picture I came across while preparing the talk. So I'd taken the title of this talk assuming that the wonderfully named Rear Admiral David Glasgow Farragut was a bit of a kind of idiot who said, damn the torpedoes, full speed ahead. But in fact, he did exactly the right thing. During the heat of battle, he climbed up the rigging to get above the smoke, and he was lashed to the rigging by his crew who thought he was going to fall off. And he had with him a pair of binoculars. And he did that, so whilst being at full speed ahead, he could actually see where he was going. So he was trying to optimize predictive validity, using my terminology. He may not have called it that, but that's what he was doing. How do you optimize predictive validity in drug R&D? It ain't easy, right? I won't for one moment pretend it's easy. You only know for sure a model is valid when it's delivered a whole bunch of drugs that work. But here are a number of things. I think if people simply recognize the quantitative importance of validity, it might help. So for example, the Medical Research Council, I guess, is the UK's equivalent of the NIH. I've done a little bit of reviewing for the Medical Research Council for grants. I can imagine, and I did it before I did this work, by the way, but I can imagine if someone had come into one of the review meetings that I was involved in, they said, look, we can do this study in 2 ways. Our gut feeling tells us that this particular method is a tiny bit better than that method, but it costs 10 times as much. So we can do one of these, or we could do 10 projects the other way using this method that we can't really prove is any worse, but I kind of guess it's a little bit worse. My guess is that the MRC would not have funded the former project, right? Not only that, certainly in the forums I was involved in, we didn't even have the information available to us, and we didn't even ask the right questions about model validity. It wasn't really part of our calculus in allocating resource, at least not in a formal way. Another thing you find is that biologists from different fields don't even use the same language, right? So if you talk to a psychopharmacologist about validity, they will use a different set of terms to a cancer biologist, right? So if you put those two folks in a room and get them to decide on projects and talk about validity, they'll probably talk about different things. So you also have to train folks to talk about and evaluate validity in a sensible way. I think another thing you do differently is I think you would invest in models, particularly public sector bodies might invest in models over and above particular therapeutics, over and above particular disease, over and above particular cures. And there's obviously some also commercial ideas here as well. So for example, if one was deploying private sector capital in a therapy area, you'd say, well, I look for therapy areas where I really think the disease models have suddenly got better. Because that's where you that's where you start deploying capital, because that's where you start making correct decisions rather than wrong decisions about what goes into clinical trials. This, I think, is grounds for optimism, and we've seen some of this at this meeting. A final concept I'll finish with, which I learned from a colleague of mine called Brendan Jackson, who was an ex-cosmologist turned bioinformatician. When he was trying to explain something that I was too stupid to understand, he said, Jack, it's all about domains of validity, as if everyone knew what domains of validity were. And apparently, all physicists know what domains of validity are. As a biologist, I've never heard of domains of validity. So if you think about Newtonian mechanics, I can't even remember all of its domains of validity, but I can remember 2 of them. Right? One is, it stops when you get to things that are only a bit bigger than an atom. Right? And then the other thing is it stops when you get near the speed of light. So Newtonian mechanics is pretty good for anything that's a lot bigger than an atom and moving a lot more slowly than the speed of light. So rockets, satellites, bridges, trains, engines, fine. Atoms and black holes, don't use it. Use something else. Physicists know that, and they apply Newtonian mechanics accordingly. I think many biologists with disease models don't even think about clearly what the domains of validity of their model are. And I think it would be interesting, for example, if people publish papers saying animal model X might be great for disease Y, reviewers should ask them to opine on the domains of validity of their model, the set of parameters under which it is valid and those it isn't. So onto the grounds for optimism. Domains of validity, I think, of models, are, in some cases, match human disease much better when you can do better patient segmentation and diagnosis. And at the start, I showed a little uptick in the number of cancer drug approvals. And I think that represents, to a large extent, the molecular slicing and dicing that goes on. So if you can identify coherent patient groups, it's much easier to match them to models where the model is genuinely predictive and has high predictive validity of the patient population in question. And that's why I think we have started to see a little uptick in drug approvals. And I think that's one of the more optimistic things I've seen at this meeting. So on that point, I'll stop.
Are there questions? Yes, here in the 3rd row.
Jack, I enjoyed your presentation. There's one topic that you did not address, and it is the ease of decision taking in organizations. And your example of the Battle of Mobile Bay is a person who can make pretty much immediate decisions and is empowered to do that and not dependent on the committee to decide whether he's moving in the right direction. Larger organizations often suffer from the inability to follow initiative because ownership is taken away for the individuals in the organization because there's always someone looking over someone's shoulder. I compare that to how a butterfly finds its mate or its food. It makes 1,000 decisions the wrong time, but it corrects quickly. If it were a farmer, it would be a long distance off from where it needs to go. So where do you place that in the context of what you have? All the other components are like hijack, overly reductionist, it's there.
Yeah.
But there is a major issue with ownership on programs and the inability of folks to listen to their data because the course is set before one knows what direction to take.
So, I mean, I think there are a huge number of practical organizational problems. I think I'll make a couple of observations. One is there is some evidence, it's not terribly strong, but there is some evidence that R&D productivity is higher in organizations of a certain size. And that certain size is not terribly small. So companies that just have got one product tend to make bad decisions because the decision is always, we should carry on investing in that product, right? And companies that are very, very big, it appears, make somewhat worse decisions. And there's a kind of middle-sized company where my guess is the senior management know enough about the technical detail of what's going on to have a sense, for example, about the validity of the scientific tools, and maybe things move a bit quicker. And then when organizations get bigger, I think you have more kind of information leakage, so possibly decision-making gets worse. That's not, I realize, addressing your question entirely about sort of different interests, right? The fact that people may have vested interests in keeping projects going and so forth. That I honestly don't know how to address. I have, however, thought a bit about this problem that I think there's a lot of implicit knowledge about validity and model quality that exists in organizations that is not made accessible to decision makers. So, for example, my sense is lots of people who work day to day with the assays or who've spent years developing the assays may know stuff about them. A lot of it's folk knowledge, effectively. It's not systematized, it's not captured. And the people making the decisions then don't have access to it. So my sense is a lot of information actually gets thrown away in bigger companies. But in terms of the interests and how you stop people having ambitions and wanting their project to go ahead, I don't know how you do that.
One closing question. Have you looked at the culture of repeat entrepreneurs successful at their mission?
No, is the answer.
Larry, you want to jump in?
Yeah, I want to ask a question. So I think yesterday, Pat Furlong and Carl Morris and I were talking about the Duchenne trials.
Yeah.
The animal models for Duchenne are awful, except for the dog model, which is okay.
Yeah.
But it means it's a bigger model. The animal takes a long time. The mouse models are bad. And we actually said what I think you said as the logical conclusion. This is a question. It will be a question in an hour. It seems to me that the logical conclusion is, this is taking what I learned so much from you here and your paper, is that you would take diseases that are awful and where people are going to die, and you would find compounds that were safe, because that's easier than finding compounds that are efficacious, and you would honor in some manner the patients and their families by saying, we don't know if this is going to work, but we'd like to do a trial of 3 people, Fred, Sally, and Jim, and we don't think this is normal, but in this situation, we have the best model, you. Is that what you said?
I think that's an extreme version of what I might say under some... Okay, no, I'll give you a short answer. So yes, right? Yes. There is a yes with provisos. There's a joke, a sort of joke, and I can't remember who said it, so I didn't think of it, but which is the best model for a cat is a cat, and preferably that cat, right? if I look at the history of drug discovery, I think one reason in the 1950s and 60s and 70s why productivity was so high was actually you almost kind of did away with the model in some cases, right? The mode of discovery in some cases was phenotypic screening in man. And it's still the case today that roughly half or more than half of all new indications of drugs are discovered in the field by observant physicians independent of the drug industry and independent of universities. And those people effectively are making observations where the throughput is incredibly low because there's only 1,200 approved drugs, right? The sample size is incredibly low because they're looking at one patient, but the measure is highly valid. Right? So that's another good sort of historical real-world example where actually validity trumps throughput. We find lots of uses for drugs when we've only got 1,200 of them on the market, and and essentially you're making observations in single people. So, and also my prejudice is that biology is pretty messy. It reuses stuff. You know, lots of drugs, if you really look at them, don't just do one thing. I think if doctors have a view about pathophysiological mechanisms, you know, sometimes these things are worth doing.
Can I say one more thing before you go? I learned, I guess I can because I got the mic, and so I learned something last week with Mark Messenbaugh at SomaLogic. We met with a guy from Medtronic, and he told us an astonishing story about the beginnings of that very large company, which I'd never heard before. The founder of that company was a kind of a mechanical guy, and he was dealing with electricity or something like that, and he was, you know, he's a smart guy, and there had been a power shortage in the hospital that he was working at, and a kid died. And the doctor who was on that kid, he died because there was no power to the thing, and it had to do with stimulating his heart. And so this guy was asked by the doctor, we can't ever let this happen again. And so the guy went back to his little shop, and he took a metronome that was about the right, you know, 1 per second something or another to make you as a pacemaker. And he rigged something that made it into a pacemaker. And he said, here, he brought it back, he said, I think this might work. This is the myth of that 80,000 employee company. And when he came back a week later to talk to the doc, it was in a patient. Isn't that an incredible story? Even if I like that story.
We have time for one more question. Yes, in the back there. Oh, we want to be closer because we've got the microphone. Sure, go ahead.
So following on from Larry's comment, this is a segue into the whole question, which I think that you, Jack, mentioned very briefly, which was the increasing regulatory, not just burden, but also ponderousness of the system. And I think that you know, the people in the pharmaceutical companies, they know the curve that you showed on your first slide. You said only 3 people in the world talk about it. They talk about it daily, okay, because this is killing them and their business. And so, you know, a good example of this is when, you know, LDL cholesterol is a bad thing. When atorvastatin was first approved, it was approved because it was safe and lowered LDL cholesterol. But some people don't respond to Lipitor, and so they need other cholesterol-lowering drugs. Now, the regulatory system is completely different. Showing that a drug is safe and lowers LDL cholesterol is insufficient for approval. Instead, you have to do a 10,000 people, 10-year study at the cost of over $1 billion to show that it prevents cardiovascular events and leads to fewer deaths. So the science isn't enough. You need to also show the other endpoint. And so this is not the only explanation for this loss of productivity, but it is a component that is substantial.
Yeah, I absolutely agree. And I think there's a number of therapy areas where you can see over time clinical trial size growing and costs going up. And also, I think one of the reasons we've had an uptick in oncology and orphan disease approvals in recent years is partly because the regulatory environment has changed. So I absolutely agree. Yeah.
Jack, thank you very much.